Well, I want to thank the organizer for putting together this interesting symposium, and I'm glad to participate and present here our recent results. So, mutation in the RYR1 gene are the most common cause of non-dystrophic congenital myopathies. The RYR1 gene codes for a protein of 5,038 amino acids that assembles in tetramers of more than 2 million Dalton that form the calcium-rich channel of the sarcoplasmic reticulum that is present in skeletal muscle, and is also known as type 1 RYR receptor. The RYR1 channels are localized at triads, or better, at the junctional domain on the terminal cisterne, from where they can contact a voltage-regulated calcium channel on the T-tubule, that is the dihydropyridine receptor, or DHPR. Activation of the DHPR by nerve-induced depolarization results through a direct interaction in the activation of RYR receptor channel and in massive release of calcium from the sarcoplasmic reticulum, and hence in activation of muscle contraction. A few additional proteins, including triadin, junctin, calsequestrin, and few others, also may either directly or indirectly interact with the RYR1 channel, and they are expected to participate in the fine-tuning of the channel activity. Mutations in RYR1 gene were initially identified in individuals susceptible to malignant hyperthermia, which is a pharmacogenetic disorder triggered by exposure to volatile anesthetics and succinylcholine. Actually, it's worth remembering that these individuals, unless exposed to a triggering event, are healthy individuals with normal muscle, both in terms of structure and function. However, shortly after, mutations in RYR1 were identified in patients with central core disease, that is the most frequent congenital myopathy, and they were also found in some other muscle disorders, collectively referred to as RYR1-related myopathies. RYR1 mutations are also responsible for some acute pathological condition triggered by heat and exercise-induced stress. These are named exertional heat stroke and exertional-induced rhabdomyolysis. These, similarly to malignant hyperthermia, occur in otherwise healthy individuals with, again, normal skeletal muscle, structure, and function. From a diagnostic point of view, RYR1-related myopathies are classified in different subtypes, mostly based on the histopathological figures observed in muscle biopsies of these patients, and these are summarized here. Accordingly, the defining histopathological feature of central core disease is the presence of a single, well-demarcated central amorphous core extending longitudinally along the muscle fibers. In multiminicore disease, the defining feature is the presence of multiple smaller cores in a single fiber. Both cores present in central core and multiminicore diseases are the results of area of reduced mitochondrial oxidative enzyme activity that is due either to mitochondrial deficiency or depletion. Nemaline bodies are found in nemaline myopathy or core road myopathy. In central nuclear myopathy, the dominant feature is the atypical presence of nuclei in the central part of muscle fibers, while in congenital fiber type disproportion, the name comes by the dimension of type 2 fibers that are at least 25% larger than type 1 fibers. In all RYR1 myopathies, there is a type 1 fiber predominance, which represents another histological finding characteristic of these diseases. At the genetic level, high number of causative mutations linked to RYR1 diseases have been identified. As shown in these slides, the mutations are clustered in three regions that are referred to as the N-terminal, central, and carboxy-terminal hotspots, which from beginning were proposed to represent regions that are critical to regulation of channel function. Indeed, recent developments in cryo-EM techniques have provided a high-resolution reconstruction of the RYR1 channel, allowing a much better definition of the structural domain within the large cytoplasmic region and in the carboxyterminal domain, where this contains the 6 transmembrane helices and the pore region of the channel. In this image, mutation due causative of diseases are shown as red dots. Alignment of causative mutations in correspondence of the structural domain identified by cryo-EM studies has revealed that several mutations are positioned near the fourfold symmetry axis at the site of interaction between the RYR1 4 subunits, or other mutations are positioned at the site of interaction between domains within the same subunit. Accordingly, the current model suggests that we can explain the effect of mutation on RYR1 function by considering that the interaction between subunits or between domains within individual subunits stabilizes the closed state of the channel, while mutation at the intersubunit interface or at domain-domain within individual subunit would induce changes that favor channel opening. At the clinical level, RYR1 myopathies are characterized by a high symptom variability. Generally speaking, RYR1 myopathies are non-progressive or slowly progressive and are characterized by the wide range of clinical symptoms, including mild muscle weakness, hypotonia, motor developmental delay, muscle stiffness, and exertional weakness that are more frequently observed. More severe presentation, including orthopedic complications like scoliosis and foot deformities, are less common, and even more rare are cases where wheelchair dependence and respiratory failure are observed. RYR variants can be inherited with a dominant or recessive transmission, and in general, there is a tendency to a more severe phenotypes observed with recessive transmission. The RYR1 mutation may either activate or inhibit channel function, or in some cases can reduce the expression levels of the RYR1 protein, resulting in reduced amount of channel in these muscle fibers. However, surprisingly, similar clinical phenotypes can result from mutation with opposite effect on RYR1 function, or in some cases, we see little or no correlation between the observed clinical phenotype and localization of mutation within the structural domains that have been identified now in the RYR1 channel structure. As an example, the same mutation can be found in some patients with a myopathy like central core disease, therefore in a patient which has muscle affected with reduced mobility, or in individual with MHS susceptibility, where these, if they are not exposed to a triggering condition, are healthy individuals with a normal life, muscle with normal structure and function. In other cases, the same mutation can be found in patients with different subtype of RYR1 myopathies, a central core, a multimini core, or other. Nevertheless, more work recently on trying to identify some form of relation between genotype and phenotype in patients with RYR1 myopathies agrees that the clinical severe cases are mostly associated with recessive inheritance or are associated with mutation localized in the cytosolic shell domain, with most of these variants being located in the bridged solenoid. In addition to the histopathological patterns that are found in RYR1-related myopathies, a different histopathological features occasionally found in skeletal muscle fibers, is represented by tubular aggregates. Tubular aggregates consist of membrane tubules that may or not contain some dense material inside, and that can be identified in a number of different muscle disorders. These tubular aggregates appear as irregular red inclusion when muscle sections are stained with a modified Gomori trichrome technique. They may stain strongly with NADH tetrazolium reductase and are negative for succinate dehydrogenase staining. These tubules staining positive when exposed to antibody against sarcoplasmic reticulum protein like SERCA. At the ultrastructural level, tubular aggregates appear as an accumulation of numerous parallel straight tubules arranged in a honeycomb-like structure in transverse section of muscle fibers at the electron microscope. The causal mechanisms that start the formation of tubular aggregates are not known, but they've been suggested to represent the response of muscle fibers to a variety of conditions, including imbalances in calcium homeostasis, metabolic alteration, protein aggregation, etc. In other words, it is accepted that they may represent a secondary, non-specific response to distressing injuries. The presence of tubular aggregates in the absence of additional histopathological feature or symptoms that may connect to any other specific muscle disorder identifies a distinct form of inherited myopathy known as tubular aggregate myopathy. And this is characterized by a muscle weakness, which is generally accompanied by increased serum creatine kinase levels, which indicate some sort of moderate fiber degeneration. Other symptoms like myalgia and cramps are often observed as secondary feature, but can also occur as isolated sign. Tubular aggregates is a rare genetic muscle disorders predominantly caused by gain of function mutation in STIM and RYR1 genes, which encode the calcium sensor STIM1 in the sarcoplasmic reticulum and the calcium channel RYR1 on the plasma membrane. A number of functional investigations using cellular and mouse models agree that mutation in STIM and RYR1 lead to an excessive cytosolic calcium level through overactivation of the store-operated calcium entry pathway. At the resting condition, when the YAR is calcium stores are full, STIM1 remains largely as a monomer, but when calcium levels are reduced and store depleted, calcium dissociation initiates a series of conformational changes whereby STIM redeploys itself, adopts a more extended conformation, and relocate to ER plasma membrane junctional site. Here, it can recruit and directly ligate the ORI channels on the plasma membrane, and this physical coupling allows entry of calcium from the extracellular environment in the cytosol. Altogether, it is accepted that alteration in store-operated calcium entry by altering intracellular calcium homeostasis are the primary cause of tubular aggregate myopathy. Although the exact mechanism that link the mutation to the assembly of tubular aggregates finally to development of myopathy are still under investigation. More recently, we identified mutation in a third gene in some patients with tubular aggregate myopathy, and this gene is calsequestrin-1. The role and involvement of calsequestrin in calcium entry regulation is supported by previous studies indicating that calsequestrin-1 has a regulatory role in regulation of store-operated calcium entry. Nevertheless, in a significant number of patients which present or have a diagnosis of tubular aggregated myopathy, no mutation in STIM or eye, or even in calsequestrin can be detected. And therefore, we are leaving these patients without a molecular genetic characterization. Accordingly, we recently started investigating if additional genes could be identified in patients with a clinical diagnosis of myopathy, but lacking a genetic diagnosis. We focused on 12 of these patients, which reported increased serum creatine kinase levels, asthenia, muscle stiffness following sustained activity or cold exposure. One of these patients had a father which reported similar symptoms. Both patients were diagnosed with tubular aggregate myopathies based on the typical presence at the histological examination of areas that were basophilic with hematoxylin eosin staining, stained bright red with Gomori, and were dark blue stained when stained with the NADH tetrazoline reductase reaction. Immunostaining revealed that these tubular aggregates were positive for SERCA, RYR1 receptor, and STIM1. Therefore, they had the hallmarks of tubular aggregates. Even more, at the electron microscope, you can see numerous tubules with a typical arrangement in this honeycomb structure, which is typical of tubular aggregates. And this is an image from patient 1. I remember again that these patients had no clinical or histological sign, additional one, suggestive of a different myopathy. So we performed next generation sequencing using a panel of genes associated to myopathy, and these resulted in the identification of causative variants in the RYR1 gene in these 2 patients. In patients 1, we identified a known causative variant or mutation in exon 40 that results in the substitution of a threonine with a methionine at codon 2206. This residue is located in the bridging solenoid 1. This mutation has been previously reported and associated with malignant hyperthermia susceptibility and is included in the list of causative mutation by the European Malignant Hyperthermia Group. In vitro functional studies have shown that this mutation induces altered functional properties to the channels by increasing sensitivity to caffeine, reducing the SR calcium stored, and increasing the resting cytoplasmic levels of the cells where this mutated channel is expressed. A second variant was also identified in patient 1, and this variant results in the substitution of a leucine with an isoleucine at codon 791. The leucine 711 islet residue is within the SPRY1 domain of the RYR1 channel. And this is a domain next, or this residue is close to the FKBP12 binding site present in RYR1. Indeed, another mutation at residue 759 was identified in a patient with central core disease, and this mutation affected the FKBP12 binding. However, the residue 7091 LEU2 islet substitution has not been clearly associated to a causative role in patients. Also, using in silico prediction program, this mutation has an ambiguous score, where according to some program, it can be probably damaging, but with other programs, the score is less severe. Therefore, in the absence of other evidences, this has to be considered as a variant of unknown significance. In patient 2, we identified another known RYR1 causative mutation that is localized in exon 45. This variant results in the substitution of a glycine with an arginine at codon 2434. And also this variant is considered pathogenic and causative for MHS as reported by the European Malignant Hyperthermia Group guidelines. Actually, this is the most frequently associated causative mutation that is found in the United Kingdom in individuals with malignant hyperthermia susceptibility. Functional studies also have shown that the GLY2434 arginine mutation results in an increased sensitivity of the channel to agonists like caffeine or 4-chlorothiazole. Also, in this patient, we identified the second missense variant. This was localized in exon 73 and results in the substitution of a glutamic acid with a glutamine at codon 3583. This residue is localized in the bridging solenoid 2. However, this variant is present with high frequency in the population, and all in silico prediction results we obtained suggest that this is a benign substitution. So there is little possibility for this variant to affect channel function. So in conclusion, causative mutation in RYR1 were identified in patients with clinical symptoms of a myopathy with an histopathology report describing the presence of tubular aggregates. These findings indicate that the number of genes that can be associated to tubular aggregate myopathy should include the RYR1 gene. Or alternatively, we can say that some form of myopathy with tubular aggregate caused by mutation in RYR1 should be included in RYR1 list of myopathies. Nevertheless, aside from this nosological classification of these two cases, certainly the results provided a molecular genetic diagnosis to these patients. The data, however, indicate that regardless of the causative mutation affecting genes that encode proteins of the store-operated calcium entry or of the excitation contraction coupling mechanism, altered calcium homeostasis represents a common central event in the development of these myopathies. However, there is an ample variability in the clinical symptoms and the histopathological feature induced by mutation in these genes. Just want to remember that the two RYR1 mutations that we identified in these two patients, which presented with tubular aggregate myopathy, these same mutations were previously found in individuals with susceptible to malignant hyperthermia, that in other words, were healthy and had no muscle normal structure and function, although they were susceptible to crisis when eventually exposed to a trigger. So even more surprising is also that these mutations were able to cause a myopathy in these two individuals and to induce the assembly of tubular aggregates in their muscle fibers, which represents an histological feature that has never been reported in a myopathy associated to RYR1 mutation. So certainly I will conclude saying that we need to identify the additional factor that modulate this variability, and that this represents certainly an important target for future research on these diseases. And with this, I want to thank the people in Siena which contributed to this work, which was essentially carried on by Daniela Rossi, Lucia Galli, and Maria Rosaria Catallo. And also want to thank Gaetano Vattemio and Giuliano Tomelleri at the Department of Neurological Neurosciences at the University of Verona. And thank you for your attention.